The standard story of human evolution runs in one direction: bigger brains, sharper tools, more complex thought. Generation after generation, for 2 million years, the hominin brain grew without interruption. Then, roughly 10,000 years ago, it stopped and reversed. The average human brain today is about 150 cubic centimeters smaller than the brain carried by people who hunted mammoths across the glacial steppe. That is approximately the volume of a tennis ball, quietly missing from every modern human skull on the planet. The reversal happened globally, across every population on every continent, right at the moment civilization was beginning. No single fossil, no single region. A species-wide turn. The fact of it is not in dispute. What caused it, when exactly the steepest phase of reduction occurred, and what it implies for human cognition are still being debated. And the debate is more interesting than any settled answer would be. To understand the scale of what happened, you need to understand the scale of what preceded it. The first members of the genus Homo appeared roughly 2 million years ago. Their brains were modest, smaller than those of later Homo erectus, which averaged around 900 cubic centimeters. These expansions correlated directly with recognizable behavioral shifts: the manufacture of stone tools, the controlled use of fire, cooperative hunting of large prey. The brain was growing because the brain was being used in new ways, and the environment was punishing anyone whose brain was not adequate to the task. By the Upper Paleolithic, roughly 100,000 to 30,000 years ago, the trajectory reached its ceiling. Neanderthals carried brains averaging around 1,500 cubic centimeters, in some individuals considerably more. Early modern humans in Europe during the same period matched or exceeded that figure. The two lineages, separated by hundreds of thousands of years of parallel evolution, independently arrived at the same approximate cranial volume. That convergence is not coincidence. It reflects the same demanding environmental pressure operating on both lineages simultaneously. The cognitive load of surviving an ice age as a generalist predator with no institutional support structure, no written records, and no specialists to defer to. That peak held through the late Pleistocene. Then, at the boundary between the Pleistocene and the Holocene, our current geological epoch, beginning roughly 11,700 years ago, the trajectory reversed. Today, the global average human cranial capacity sits at approximately 1,350 cubic centimeters. The loss of 150 cubic centimeters over 10 millennia is not dramatic in any single generation. Across the full span, it amounts to a 10% reduction in brain volume. In evolutionary terms, that is fast. 2 million years of growth, unwound by a tenth in less than 12,000 years. The metabolic cost of maintaining a large brain makes the reversal more striking, not less. Brain tissue is among the most energetically expensive tissue in the body. The modern human brain, at approximately 2% of total body weight, consumes roughly 20% of resting caloric expenditure. That ratio is extraordinary by mammalian standards. Evolution does not maintain expensive organs without an immediate survival return. For 2 million years, the return on that investment was clear enough to justify continuous expansion. Something changed that calculation, and it changed it globally, across every human lineage on the planet, within a geologically brief window. The question is not whether it happened. The fossil record is clear on that. The question is what set it in motion, and whether the four major candidates are separate explanations or pieces of the same mechanism operating across different timescales and through different biological pathways. Before the hypotheses, the methodology, claims about ancient brain volume are only as good as the measurement techniques behind them. For most of the 19th and early 20th centuries, paleoanthropologists measured cranial capacity by plugging the natural openings of a fossilized skull and filling the interior with a known material, mustard seeds, lead shot, or water, then decanting that material into a graduated cylinder. The method worked well enough to establish the broad pattern of hominin brain expansion, but it introduced measurement error and occasionally risked damaging irreplaceable specimens. Current practice relies primarily on micro-CT scanning, A fossilized skull is passed through a computed tomography scanner that generates thousands of cross-sectional X-ray images at high resolution. Software assembles these into a three-dimensional digital model of the void inside the skull, a virtual endocast. Volume measurement from a virtual endocast is precise to fractions of a cubic centimeter. The inner surface of the model also preserves impressions left by the brain's folds and blood vessels on the bone, providing limited but real information about brain organization, not just size. Raw volume, however, is not sufficient on its own. A blue whale's brain weighs close to 9 kilograms. Elephants, sperm whales, and Neanderthals all have or had larger absolute brain volumes than modern humans. Brain size scales with body size because a larger body requires more neural tissue simply to operate its musculature and process sensory input across a larger surface area. The encephalization quotient, EQ, corrects for this by comparing an animal's actual brain mass against the predicted brain mass for an animal of its body size. Humans have an exceptionally high EQ. The critical finding for the Holocene shrinkage story is that when researchers apply EQ analysis to the post-Pleistocene data, the brain shrank faster than the body. Our bodies did get smaller after the Ice Age, but the brain outpaced that reduction. Something was targeting neural tissue specifically. The timeline of that targeting is where the science becomes contested. A 2021 study by De Silva and colleagues, analyzing a large data set of skulls spanning the full period, proposed that the sharpest reduction occurred not at the agricultural transition, but around 3,000 years ago, coinciding with the rise of large, literate, bureaucratically organized societies. In 2022, a separate team reanalyzed the same dataset and reached a different conclusion. No sharp recent drop, but rather a slow continuous decline extending back 30,000 years, with gradual acceleration near the agricultural boundary. Same fossils, different statistical framing, different conclusion. The disagreement is not a failure of the science. It is a demonstration of its actual difficulty. The number of well-preserved skulls with reliable dating spanning the full Pleistocene to Holocene transition is finite and smaller than most people assume. Depending on which specimens are included, how outliers are treated, and how the statistical model is constructed, the curve shifts. What both teams agree on is the direction of travel. The brain shrank. When the sharpest phase of that shrinkage occurred remains an open question, and the answer matters, because it changes the weight assigned to each of the four pressures below. The transition out of the last glacial maximum into the early Holocene was not a gradual, gentle warming. It was punctuated, rapid in geological terms, and accompanied by massive reorganization of terrestrial ecosystems. Ice sheets retreated, megafauna populations collapsed. The cold, dry, open steppe environments that had characterized much of the northern hemisphere for tens of thousands of years gave way to warmer, wetter, more heavily forested landscapes. Bodies respond to sustained thermal environments through mechanisms described by two related biological principles. Bergmann's rule holds that animals in colder climates tend toward larger, bulkier body mass, because a larger mass has a proportionally smaller surface area, which slows heat loss. Allen's rule describes the complementary pattern in limb and appendage proportions. Cold-adapted populations have shorter, thicker limbs that retain core heat, while warm-adapted populations have longer, thinner limbs that radiate it. Both patterns are well documented across mammal species and confirmed in comparative studies of human skeletal remains across different climatic zones. As the Holocene warmed, selective pressure on body mass shifted. Carrying excess metabolic bulk in a temperate or tropical environment was a thermoregulatory liability rather than an asset. A cold-adapted brain, large, spherical, generating heat continuously, had functioned as a useful contributor to core temperature in a freezing environment. In a warm one, sustained heat production from a massive organ created a cooling problem that a smaller, leaner brain did not. The skeletal record confirms that post-Pleistocene human bodies did in fact shrink. Average height dropped, Limb proportions shifted in ways consistent with adaptation to warmer conditions. The brain reduction that accompanied this process was real, but as EQ analysis established, the brain shrank faster than the body. Temperature-driven body mass reduction explains part of the brain reduction. It does not explain all of it. Climate set the stage and contributed to the outcome, but it was not operating alone. The timing of the agricultural transition and the timing of the initial brain shrinkage overlap closely enough that the connection is hard to dismiss, but the mechanism is not what most people assume. Agriculture did not make humans better fed. It made humans more consistently fed on a narrower and nutritionally inferior diet, while simultaneously exposing them to a new class of infectious disease. Early farming economies across the Fertile Crescent, the Yangtze River Basin, and Mesoamerica converged on a small number of high-yield starchy staples: wheat, barley, rice, maize. These crops could support dense populations. They could not, on their own, support well-nourished ones. The contrast with the preceding forager diet is measurable. Hunter-gatherer populations consumed a diverse array of wild plants, animal proteins, and dietary fats. The nutritional breadth available to a mobile forager band moving through a varied landscape, tubers, seeds, leafy plants, insects, fish, game, is substantially wider than what a sedentary farming family, subsisting primarily on grain, could access. Archaeological evidence of forager health, read from their skeletal remains, shows generally robust bone density, fewer metabolic stress markers, and greater average stature than their early agricultural counterparts. The skeletal record of early agricultural populations is a catalog of nutritional stress. Harris lines, horizontal bands of increased bone density visible in cross-section, record episodes of severe childhood malnutrition or illness during which bone growth temporarily halted. Dental enamel hypoplasia, which marks the same growth disruptions in tooth development, follows the same pattern. Average stature dropped significantly at the agricultural transition in multiple independent regions, and recovered only slowly over subsequent millennia as agricultural systems diversified and improved. Living in close proximity to domesticated animals introduced a further burden. Zoonotic diseases, illnesses that jump from animals to humans, require dense, sedentary human populations near animal populations to establish themselves. The earliest farmers provided exactly that. Measles, smallpox, influenza, and tuberculosis all have animal reservoir origins. Communities that had rarely experienced epidemic disease became susceptible to it. The brain is the body's highest maintenance organ. During early childhood, when neural architecture is being established, it demands high-quality protein and dietary fat, particularly omega-3 fatty acids. A child developing on a primarily starchy, protein-deficient diet, whose immune system is simultaneously fighting novel infections, cannot fully fund both neural development and immune function. The body triages. In environments where adequate nutrition was structurally unavailable, a massive, energy-hungry brain became a lethal liability. Evolution ruthlessly selected for efficiency. Individuals with genetically slightly smaller, less metabolically demanding brains were more likely to survive childhood famines and reproduce. Over thousands of generations, the average cranial capacity dropped. The self-domestication hypothesis begins with an observation that is easy to verify and difficult to explain away. Every animal humans have domesticated has ended up with a smaller brain than its wild ancestor. Dogs have brains roughly 20% smaller than wolves of equivalent body size. Domestic pigs have smaller brains than wild boars. Domestic cattle have smaller brains than aurochs, their wild progenitor. The pattern holds across species, across continents, across thousands of years of independent domestication events carried out by human cultures with no contact with one another. Whatever domestication does to an animal, brain reduction is a consistent outcome. The clearest experimental evidence for the mechanism comes from Soviet geneticist Dmitry Belyaev, who in the 1950s began a selective breeding program with wild silver foxes at the Institute of Cytology and Genetics in Novosibirsk. The selection criterion was simple: tameness. Foxes that tolerated human approach without aggression were preferentially bred. Foxes that bit or fled were culled from the breeding pool. No selection was applied to anatomy, coloration, or any physical trait. Within a few generations, the foxes began exhibiting a suite of physical changes that no one had deliberately selected for: floppy ears, curled tails, patchy pigmentation, shorter snouts, and reduced cranial volume. The behavioral selection had altered developmental pathways that controlled traits no one was selecting for at all. A single target produced an entirely different animal. This suite of unintended physical changes accompanying selection for tameness is called domestication syndrome. The pattern itself is well documented across every domesticated species. Selecting for one behavioral trait alters developmental pathways that govern many unrelated physical traits simultaneously. The brain is among the structures those pathways shape. What Belyaev demonstrated in a controlled setting over a few decades, nature appears to have run across millennia on a far larger population, humans living in increasingly dense, socially complex settlements. The application to humans is a hypothesis, not a confirmed finding, but its internal logic is consistent with the animal evidence. The transition from bands of 50 or fewer highly mobile foragers to settled villages of hundreds, then towns of thousands, then cities of tens of thousands, created a new and intense selective environment. Reactive aggression, the kind that produces impulsive violence, becomes increasingly costly in dense settlements where retaliation is swift and collective punishment for antisocial behavior is possible. Individuals capable of suppressing reactive aggression in favor of negotiation, deference, and cooperative tolerance survive and reproduce at higher rates in that environment. Those who cannot are removed from the gene pool through execution, exile, or social exclusion over generations. If the mechanism is analogous to what Belyaev documented in foxes, and the anatomical changes associated with human domestication syndrome, including reduced brow ridges, smaller faces, and reduced tooth size, do appear in the skeletal record across the Holocene, then brain reduction may have arrived not as a direct target of selection, but as a developmental byproduct of selecting for a more cooperative temperament. In 2021, the same year De Silva and colleagues published their timeline analysis, a separate team published a study on ant brain size across species with different colony structures. The finding was straightforward. Ant species that live in large, complex, highly organized colonies have smaller individual brains than ant species that live in small, loosely structured groups. The cognitive load carried by the individual is lower when the colony handles more of the decision-making collectively. Evolution charges for what it builds and discounts what is no longer needed. The parallel to humans is the most structurally ambitious of the four hypotheses, and the most dependent on the timeline question left open in Chapter 2. If De Silva's reading is correct, and the sharpest brain reduction occurred around 3,000 years ago, then the most powerful explanatory pressure is not agriculture or climate or domestication. It is the emergence of externalized knowledge storage at civilizational scale. A Pleistocene hunter-gatherer in a band of 30 to 50 people was a generalist in the most demanding sense possible. There were no specialists to defer to, no written records to consult, no institutional memory beyond what the oldest members of the group could carry in their heads. Every adult had to independently hold functional knowledge of local plant toxicology, animal behavior, seasonal water sources, navigation across vast unmarked terrain, weather pattern recognition, tool manufacture, and wound treatment. This was not a division of cognitive labor. It was the full weight of survival knowledge carried simultaneously by every adult in the group. Forgetting any part of it carried direct survival consequences. The 1,500 cubic centimeter brain was, in part, the hardware required to run that load reliably without any external storage at all. Agriculture broke that cognitive monopoly. Surplus calories freed some people from subsistence tasks entirely. Specialization became possible, then inevitable. A potter did not need to know how to track aurochs. A soldier did not need to know which roots were edible in autumn. A scribe did not need to know how to knap flint. Knowledge moved out of individual heads and into the social fabric. Oral tradition, apprenticeship, craft guilds, administrative records, and eventually writing Each transfer of knowledge from individual memory to external storage reduced the selective premium on large individual brains. The transition was gradual and cumulative. Oral tradition alone, specialized knowledge held by designated members of a community and transmitted through structured narrative, represented a form of collective memory that no single brain needed to replicate in full. The emergence of writing accelerated the process dramatically. A written record does not forget. It does not misremember seasonal patterns or contract quantities. It does not die and take irreplaceable knowledge with it. The cognitive functions that writing absorbed from individual memory were precisely the kinds of high-volume, precision recall functions that most demanded raw neural capacity. This is not a metaphor. It is a claim about where cognitive work was being performed and what happened biologically when the distribution of that work shifted. An individual embedded in a complex, literate, specialized society does not need to be a walking encyclopedia to survive and reproduce. Evolution, which does not commit to maintaining organs beyond their survival value, responded accordingly. The brain that no longer had to carry the full cognitive burden of an isolated individual gradually shed the mass that burden had required. The question the evidence inevitably raises, whether a smaller brain means diminished capability, rests on an assumption that the data does not support. Absolute brain volume is a poor proxy for cognitive performance. Elephants have brains averaging 5 kilograms. Sperm whales carry brains of up to 9 kilograms. Neither species has demonstrated calculus or built written language. Volume alone predicts very little about what a brain can do. The cerebral cortex, the heavily folded outer layer of the brain responsible for most higher cognitive processing, does not scale linearly with overall brain volume. The brain increases its effective cortical surface area by folding. The grooves are called sulci, the ridges gyri. A smaller brain can maintain substantial functional surface area through denser folding. The research packet does not suggest that the Holocene reduction came with any measurable loss of cortical complexity. Synaptic pruning works the same way at the individual level. During childhood brain development, the brain initially overproduces neural connections, far more synapses than the mature brain will use. Beginning in adolescence, it systematically eliminates unused connections, strengthening frequently used pathways and discarding redundant ones. This pruning is not a loss of capability. It is a gain in processing efficiency. Shorter, more direct signal pathways operate faster than sprawling networks carrying redundant connections. The brain improves through selective reduction, not through accumulation. A mature adult brain is more capable than the same brain at the peak of its synaptic density in early childhood, not despite the pruning, but because of it. The evolutionary shrinkage over the Holocene may reflect an analogous process at a longer timescale. The raw volume required to run a lone generalist survival encyclopedia to maintain the full cognitive apparatus of an isolated Ice Age individual was genuinely substantial. As collective intelligence absorbed more of that load, the individual brain no longer needed to fund all of it internally. What remained was not a diminished brain. It was a leaner one, operating within a larger system whose total cognitive capacity dwarfs anything a single skull could contain. The brain did not shrink because humanity failed. It shrank because humanity built something larger than any individual brain, and then offloaded onto that structure the cognitive overhead that individual brains had been carrying alone for 2 million years. The tennis ball of missing brain tissue is not a monument to decline. It is a record of what civilization cost and what it replaced. Whether the shrinkage has continued, slowed, or stabilized in the most recent centuries is not yet resolvable from the available data. The fossil record thins out precisely where contemporary history begins, and the kinds of systematic cranial measurements that would settle the question require sample sizes and methodological consistency that have not yet been achieved across modern populations. What is clear is that the pressures which drove the reduction, dietary change, social complexity, dense collective living, the ongoing expansion of externalized knowledge systems, have not reversed. The conditions that made individual cognitive bulk less necessary have only deepened. Agriculture became a global food system. Oral tradition became libraries. Specialization became entire professions built around single, narrow functions. If anything, the conditions that made a large individual brain less necessary in 3000 BCE are more pronounced now than at any prior point in human history. The answer to whether the brain is still shrinking may simply be there is no reason to expect it to stop.